Hello class, so we will be going over cellular respiration and mainly focusing on oxidative phosphorylation, um, which is composed of the electron transport chain and chemiosmosis. I thought it was important to point out that I will not be mentioning these terms here because these happen in the previous steps of cellular respiration. So we start off with glucose in glycolysis. So it's basically just splitting up the glucose into 2, 3 carbon molecules, which is pyruvate. As well, we will go ahead and not mention acetyl CoA because that happens here in these steps in pyruvate oxidation, and that's what starts off our citric acid cycle. We will also now be going over carbon dioxide in my model because that happens in the citric acid cycle and we don't see it in oxidative phosphorylation. This CO2 is made as a byproduct by citric acid and is essentially what we exhale out. As well, I would now go over substrate level phosphorylation because that happens in these 3 steps and not in the electron transport chain and chemiosmosis, which is oxidative phosphorylation. Okay. So I thought I would show you a little mitochondria here to show us where the electron transport chain and chemiosmosis happen. So here we have our intermembrane space, our mitochondrial matrix. So this is our intermembrane space here, and our mitochondrial matrix down here. And the inner membrane is where our proteins and ATP, our proteins, the complexes in our ATP synthase are placed. So we know that we start off the electron transport chain with 10 total NADH, 2 from the glycolysis, 2 from pyruvate oxidation, and 6 from NADH. 6 NADH from the citric acid cycle, and then also 2 of ADH that come from the citric acid cycle. So we have our little NADH little electron carriers coming over from the other steps and arrive at the mitochondrial matrix. And so the NADH drop off their electrons and it's oxidized back to NAD plus, and it goes back to get more electrons from the other steps. And so we have our complex 1, 2, and 3, and 4 here, which go ahead and do a series of reductions and oxidations, and it makes our now high energy electron move down the electron transport chain. We have as well these little like ferries that go ahead and drop off our electrons from one complex to the other. So we see here that FADH doesn't start at the complex 1 like NADH does. So we don't. So we say that FADH does not release as many hydrogen ions into the intermembrane space because it starts off a little later and comes into the its electrons come into complex 2. So our electron moves down the electron transport chain, and when it does, it releases hydrogen ions, essentially, which are essentially protons, into the intermembrane space. At the end, the last complex 4, which is the last electron acceptor, gives off its electron to a water molecule. So right here, and so since oxygen is highly electronegative, it also pulls some hydrogen ions, which create H2O, so water. So eventually we have all these little hydrogen ions up here, and they are trying desperately to go down their gradient, their concentration gradient, but they have nowhere to go except through ATP synthase, which provides a channel for them to go down into the mitochondrial matrix. And when this happens, it powers ATP so that it can be phosphorylated into ATP. And that's how we go ahead and create ATP essentially. So ATP synthase is described as a like a water wheel, and the hydrogen ions are described as water, and that sort of helped me wrap my head around what it does, or like how ATP synthase, how it works, as well. The spinning motion of the ATP synthase rotor is what is what powers ATP and gives energy to phosphorylate ADP into ATP, and that's how we create our ATP. So thank you.